Hello friends, today we are going to see plasmids as a vector. In that, we are going to see PBR322 plasmid vector. First we will see the introduction about molecular cloning and then about vectors. The basic procedure of molecular cloning involves a series of steps. First, the vector and the DNA fragments to be cloned are generated by using the same restriction endonuclease enzymes. Second, vector and DNA fragments produced by digestion with restriction enzymes are ligated to each other by the enzyme ligase. Third, this recombinant DNA molecule is transferred to a host cell where it replicates and produces large number of identical copies known as clones. Finally, the cloned DNA segments can be recovered from the host cell, purified and analyzed in various ways, and are also used to produce transgenic plants or animals. Gene cloning deals with the transfer of desirable gene into a host cell. The cell which receives desired gene is called host cell. The desired gene cannot be directly introduced into a host cell because the desired gene does not carry the origin of replication. Hence, the desired gene has to be introduced into a autonomously replicating extrachromosomal bodies, and these extrachromosomal bodies are present in bacteria, bacteriophages and viruses. These extrachromosomal bodies transport the desirable DNA fragment into a host cell, and thus they are termed as vectors or cloning vehicle. So we can say that vectors are self-replicating DNA molecules used to transfer foreign DNA segments between host cells. Now, what are the properties of a good vector? The vector should be small in size. It must be able to replicate in the host cell, that is, it must have an origin of replication. It must have unique restriction site for a particular restriction enzyme. Vectors must contain a selectable marker that allows easy detection or selection of transformed bacterial colony. It must have multi-cloning sites or polylinker. It should contain specific control systems like promoter, terminators, ribosomes, binding sites, etc. so that the cloned DNA should express properly. As a molecular tool, various cloning vectors are currently being used in genetic engineering, which are plasmids, phagemids, and cosmids, etc. Now let's see plasmids as a vector. Plasmids are extrachromosomal, double-stranded, circular, self-replicating DNA molecules. The plasmid performs several functions, including resistance to antibiotics. Almost all bacteria have plasmids containing low copy number and high copy number. The term plasmid was originally introduced by Lederberg in 1952. Bacteria are the common source of plasmids. In 1973, Stanley Cohen and co-workers first reported the use of bacterial plasmids as molecular cloning vectors. Now let's see the characteristics of plasmid. It must have minimum amount of DNA. It must have relaxed replication control. It should have at least two suitable markers for identification. It should possess a single restriction site for at least one restriction enzyme. The unique restriction site must be located within one of the two selectable markers. The plasmids that occur naturally do not possess all the characteristics to be used as cloning vectors. Therefore, plasmids must have been artificially developed by modifying naturally occurring plasmids by in vitro techniques. Some of such plasmids are PBR322, COL-E1, PUC, etc. PBR322 is the first artificially prepared plasmid commonly used in genetic engineering. It is derived from E. coli plasmid and engineered into a cloning vector. Let's see PBR322 as a plasmid vector. It is an ideal cloning vector most widely used in genetic engineering derived from E. coli plasmid. PBR322 is a reconstructed plasmid. In PBR322, the first letter P denotes plasmid and BR indicates the name of two scientists, Bolivar and Rodriguez, who constructed this plasmid. 322 is a number given to differentiate it from other plasmids. It consists of 4363 base pairs and consists of a region of replication which allows production of multiple copies per cell. It also possesses genes which confirms resistance to two antibiotics, namely ampicillin and tetracycline, and unique recognition site for total 20 restriction endonucleases. The tetracycline gene has 8 restriction sites and the ampicillin gene has 3 restriction sites. The gene for tetracycline contains unique recognition sites for Hint3, BAM-H1 and CEL1. Similarly, ampicillin resistance gene contains PIST1 site. Now this is the physical map of plasmid PBR322. Here you can see the resistance gene tetracycline having restriction site Hint3, BAMH1 and CEL1 and the other ampicillin resistant gene having the endonuclease restriction site PIST1. This plasmid is also having an origin of replication. If any foreign DNA molecule is inserted at any of these 11 sites, insertional inactivation of any of the antibiotic resistant markers takes place. The presence of two antibiotic resistant genes are beneficial during selection and screening process. The presence of restriction sites within the marker permits an easy selection of transform cells as the insertion of DNA fragment within any of the marker gene makes it non-functional. The antibiotic resistant cells can be selected by culturing the cells on nutrient medium supplemented with ampicillin or tetracycline, depending upon which gene is disrupted or damaged during insertion of target DNA. For example, if it lies within tetracycline gene, the cells that do not grow on tetracycline medium are sensitive to tetracycline and actually carry our cloned DNA. This process allows an easy selection of a single bacterial cell having recombinant DNA. The screening is done in two steps. First, the transformed cells are grown in ampicillin containing medium. Only transformed cells containing ampicillin-resistant genes are able to grow. In the subsequent step, the cells that are grown on ampicillin media are transferred to a tetracycline containing media. In transformed cells, the tetracycline gene is disrupted or damaged due to insertion of target DNA, as it lies within this gene. The cells do not grow on tetracycline medium, are sensitive to tetracycline, and actually carry our cloned DNA. Significance of PBR322. Its small size enables easy handling, purification, and manipulation. Two selectable markers, ampicillin and tetracycline, permits easy selection of recombinant DNA or transformed cells, which can be selected by growing them on subsequent media. About 15 copies of them occur per cell, which can be increased to 1000 to 3000 when protein synthesis is blocked. It can be used for construction of other plasmids. Hope you understood the topic very well. Thank you.